Drought-stress plants are subjected to a variety of environmental challenges as they grow and develop in both natural and farmed settings. Drought is one of these stresses, which is characterized as the primary environmental stress caused by temperature fluctuations and light intensity. Also has a negative impact on plant morphological, physiological, biochemical, and molecular characteristics, as well as photosynthetic capacity. The drought unpredictable nature is due to a number of variables, including the uneven and unreliable distribution of rainfall, evapotranspiration, and water storage capacity across the rhizosphere. These elements have a negative impact on plant production and quality. Plant responses to drought are influenced by growth stages, age, plant species, and the intensity and duration of the drought. Drought stress is one of the most important environmental stresses and occurs for several reasons, including low rainfall, salinity, high and low temperatures, and high intensity of light, among others. Drought stress is a multidimensional stress and causes changes in the physiological, morphological, biochemical, and molecular traits in plants. Many plants have improved their resistance mechanisms to tolerate drought stress, but these mechanisms are varied and depend on the plant species. Effects of drought on plants. The effects of drought range from morphological to molecular levels. These changes can appear at all phenological stages of plants' growth at whatever stage the water deficit takes place. Crop growth and yield. The most important effect of drought is that the germination and seedling stand are inhibited. Water relations. There are some important characteristics that can impact water relations: relative water content, leaf water potential, stomatil resistance, rate of transpiration, leaf temperature and canopy temperature. Nutrition uptake. Drought and limitation in water availability influence the nutrients uptake and their diminished tissue concentrations in crop plants. Another important effect of drought is on the acquisition of nutrients. The root and their transport to shoot. Oxidative damage. Exposure of plants to certain environmental stresses quite often leads to the generation of reactive oxygen species. Reactive oxygen species may react with proteins, lipids and deoxyribonucleic acid, causing oxidative damage and impairing the normal functions of cell. Drought resistance is mechanisms causing minimum loss of yield in a drought environment. Different mechanisms through which a crop is capable of minimize the loss in yield due to drought stress are grouped into three categories: 1. Drought escape, 2. Dehydration avoidance, 3. Dehydration tolerance. Drought escape describes the situation where drought-susceptible variety performs well in a drought environment simply by avoiding the period of drought. Early maturity is an important attribute of drought escape, suitable for environments subjected to late-season drought stress. Early varieties usually contain lesser leaf area index, minor total evapotranspiration and inferior yield potential. Dehydration avoidance. It is the ability of a plant to retain a relatively higher level of hydration under conditions of soil or atmospheric water stress. Dehydration avoidance can be achieved either by reducing transpiration, water savers, or increased water uptake, water spenders. The following plant characteristics are responsible for dehydration avoidance. I. Reduced transpiration by closure of their stomata in water-saving xerophytic species in response to water deficit well before wilting. In less irrigate plants, stomata possibly will remain open during morning hours and close as solar radiation increases. In cam plants, stomata closes during the night, when CO2 is fixed by them. 2. Osmotic adjustment is an important mechanism positively affects growth and yield under stress. 3. Increased concentrations of abscisic acid stress hormone plays an important role in water stress avoidance by affecting stomata closure, reduction in leaf expansion and promotion of root growth. Many of stress-responsive genes encode such proteins, for example, dihydrin, osmotin, Li proteins etc. are also produced in response to ABA. Genes encoding such proteins are called ABA-responsive, ABAR, genes. 4. Deposition of wax within and over the cuticle reduces transpiration during water stress. V. Leaf characteristics like leaf pubescence, erect leaf angle, leaf rolling etc. Vi. Increased water uptake by deep root system. Dehydration tolerance of a genotype means that a significantly lower level of changes in it than those in another genotype when both of them are subjected to the same level of dehydration. Various measurements of dehydration tolerance are as follows. I. Maintenance of membrane integrity by low levels of solute leakage. 2. Seedling growth parameters like seedling survival or recovery, seedling growth, seed germination under osmotic stress created by polyethylene glycol, PEG, stem reserve mobilization, presence of ONs in cereals, high levels of proline accumulation. Plant drought response mechanisms and main related traits.